Certainly a pleasure to be back here again. I always enjoy this meeting. What a great caterer. Let's give the caterers a hand. I always go home a couple pounds heavier. My doctor hates to hear that. But I want to just share with you a little bit of research that we have ongoing at the research station tonight, and then we'll open up for questions. If you've got a question as we're going along, please ask it, and I'll be happy to answer. I brought the wrong file tonight, so there's a couple slides missing, and I'll just tell you about them at the end because it's some pretty interesting data from a grazing study that we conducted this summer at the research station and last summer. All right, and my contact information's on there. If you ever have questions, please feel free to call me. I'm always happy to visit with you, and you can go through Todd too. It's a great agent. First thing I want to talk a little bit about is summer annuals, and I've been doing work with these for a number of years. Actually, I've been testing them at the research station since the early 2000s when I came, and they've got a place in grazing systems. There shouldn't be a grazing system themselves, but they've got a place, and that place is really supplying forage during the summer months when we need a high-quality forage. The advantages of using annuals in grazing systems is they tend to be fast germination, rapid growth, high productivity, and good quality, and that can put gains on your calves. If you're weaning calves and you want something to go on to, an annual is a great thing to go on to with the weaned calf because it's just high in quality, and those calves are going to do well in that. If you're grass finishing or have some kind of a combination of grain and grass, incorporating some annuals into that system would be really good in terms of keeping the average daily gains up on those calves. The biggest disadvantage is you got to establish them every year, and that costs money, and we figure somewhere around $150 to $175. And that sounds like a lot of money, but if you manage them right, you're going to get that back and more from these annuals. So this is kind of my take on annuals and forage systems. Profitable forage systems are going to be based on a well-adapted perennial sod. In our part of the country, that's going to be tall fescue. And then that sod's going to be supplemented with annuals to provide forage when we need them in that system. So I want to just spend a few minutes and talk about summer annuals that we've been working with at the research station for a number of years. And this is from a field day we had a couple years ago at Robert Harper's farm up in Goochland. And he was grazing some brown midrib sorghum Sudan grasses, kind of strips of some different varieties. And we had a great field day. That was the hottest field day I've ever been at, 105 degrees. We had over 200 people sign up for the field day. 150 showed up even in that heat. And that was one to remember. So this is kind of where those summer annuals fit into a grazing system. The green on this graph is the tall fescue curve here. And we got good growth in the spring. We got a second hump of growth in the fall, but not much going on in the summertime. And that's where these summer annuals really fit into grazing systems at. And they can provide a plentiful and high-quality feed source during the summer months. One thing people don't realize about cool season grasses like orchard grass and tall fescue, even with adequate moisture in the summertime, when we get warm temperatures, we're talking 90 degrees during the day and stays up around 80 at night or a little bit cooler, growth on those grasses are limited by temperature, even if they have adequate moisture. So it's important to remember that summer annuals, in contrast, they're not going to reach optimal growth until around 90 to 100 degrees Fahrenheit. So they have the potential to grow if they have a little bit of water in the summertime. So when we recommended summer annual varieties in the past, this was the old recommendation. We find a reasonably priced, locally available variety and focus on management. And certainly management with summer annuals is important. I can't stand driving past a field of summer annuals in the summertime and they're out there and they look decent, but they're just yellow because we didn't put any nitrogen fertilizer on them. If you're going to have summer annuals, you've really got to manage the fertility and manage the grazing to get the most out of those summer annuals. Now, this is may have been a good variety back in the older days, but we've had some real advances in breeding programs with some of these summer annuals. Things like the brown midrib trait, that's called BMR trait. The brown midrib trait is associated with increased digestibility in these summer annuals. They have lower levels of lignin, and that translates into higher animal performance. And as I said earlier, we've been conducting variety trials since the early 2000s, but it wasn't until more recently that we started to look at more than yield. We started to look at digestibilities in these summer annuals. We've evaluated at the research station lots of different ones: sorghum Sudan grasses, pearl millets, teff, crabgrass are all summer annuals we've evaluated over the years. And pretty standard nitrogen management. They get about 75 pounds of nitrogen at seeding, and then about 60 pounds after each cutting in this harvesting system. If you were in a grazing system, you'd put on about 60 and then about 40 after each grazing to keep production up. This was the first year that we started to evaluate digestibility in this trial, and this was the first harvest in 2009. And what's interesting about this, and we've got some different species in here. We've got forage sorghum, we got sorghum Sudan grass, pearl millets in this trial. Some had the BMR trait, some did not have the BMR trait. When we looked at yield at the first harvest, it was pretty amazing at the range in yield. So these were all planted at the same time and harvested at the same time. The range in yield was 3,800 pounds at that first harvest to 6,800 pounds. Tremendous difference in yield. And then we got the data back from the lab that looked at IVTD. That stands for in vitro true digestibility. That's when you take that plant and you put that plant tissue and rumen fluid from the animal that you get out of a hole in the side of the cow. You actually dip some rumen fluid out and put the forage in that and measure what the rumen microbes digest in that forage. And that gives us a little better estimate of how digestible that forage is going to be in the animal's digestive tract. So when we looked at that, the range in this in vitro true digestibility was 54 to 74%, so a huge difference in digestibility. You know, when I saw those numbers come back from the lab, I said, well, you know, the ones that yield the most are going to be lowest in digestibility. And we started to match those numbers up, and lo and behold, some of the ones that yielded the most were some of the most digestible in the trial. And that really kind of got me interested in this relationship between yield and digestibility. And we've been looking at that relationship over the last 5 years or so. So this was in 2009. And what this graph is on the bottom we have yield, and range 2 to 10,000 pounds. And then on this axis here, the y-axis, we have in vitro true digestibility. And we graph these two against each other. And in 2009, there was no relationship. It looked like somebody just shot that graph with a shotgun. And that kind of got me interested. The ones that I'd really like to have are the ones that yield a lot, right? And have good digestibility. And we've looked at this relationship over the last 5 years. And some years we have a relationship like there. This was 2011. And we had, as the yield increased, our digestibility tended to increase in 2011. And I'm not going to show you data from all the years, but I'll summarize it here. 2009 and 10, we had no relationship at all between yield and digestibility. In 2011, 12, and 14, we had a negative relationship. So as digestibility increased, yield tended to decrease. 2014 was a really weak relationship. These were fairly strong to moderate relationships in 2011 and 12. So what's causing this? We don't completely understand that. It's probably weather related. It seems like in years where we have really high yields, we tend to have a stronger negative relationship. But we don't fully understand all the factors that it's impacting this relationship, and it does fluctuate from year to year. But the point that I want to drive home tonight is that we've got to look for these outliers. We've got to look for these varieties that have both high yield and high digestibility, and those are the ones that we really want to put into forage programs. One thing that's been consistent over this 5-year period is that if you choose varieties to include that have the BMR trait, the brown midrib trait, they're more digestible. This is on average. Each year we have somewhere between 20 and 30 varieties in this trial. And every year of the trial, you know, we see a 5% difference on average between a BMR, which is the black bar, and the gray bar, which is the non-BMR varieties. Some of the BMR varieties are much more than 5% digestible, sometimes up to 15% more digestible. And some are a little bit less. So there is some variation even within the BMR trait in terms of digestibility. But if you just go and average all this over all the BMR varieties and all the conventional varieties over the whole trial, we usually see at least 5% difference in digestibility. And that translates into improved performance in the animals grazing that. So I've got a lot of tables like this, and nobody likes to look at these kind of tables. And there's a lot of data in here. And what I did was I went through and kind of summarized these trials and ranked these varieties by yield. And a lot of times when I show people Excel spreadsheets with all this data in it, their eyes kind of roll back and they kind of shut down. And I have tried to figure out a better way to show people what this table is really trying to tell us. And so what we've done is we've taken all that data in that table and we've graphed yield against digestibility, but we index the yield against the average for the trial. So what's this mean? On the bottom here where it says 0, this is the average yield for the trial. So everything that yielded above average is over here. Everything that yielded below average is over here. We did the same thing on the y-axis with digestibility. So everything that's above average in digestibility is up on the top of the graph. Everything that's below average in digestibility is on the bottom of the graph. And this is a really nice way to look at these varieties because it makes 4 nice quadrants in this graph. We have varieties in this bottom, it'd be your bottom left-hand quadrant. They have lower than average digestibility and lower than average yield. Those are varieties I really don't want to put into a grazing system. Those are the ones that we want to avoid. Sometimes knowing what to avoid is as important as knowing which ones to choose. And then we've got varieties up here in this quadrant. They have above average yield and above average digestibility. These are varieties that we want to think about incorporating into a grazing program. They've got good yield and they've got good digestibility. And those are the ones that we have to look for as we go through all this data. So what I've done is I put all these varieties that had above average yield and above average digestibility into a table. And then we went through and we started to look for varieties that had above average yield and above average digestibility for more than 1 year. And what we came up with was 3 out of 5 years had above average yield and digestibility. What's that mean? That means those are pretty robust varieties. So they've done well over multiple years. Those are ones that you may want to think about putting into a forage program. Alta Seeds has 2 varieties. This 9301 happens to be a Sudan grass hybrid, and this is a sorghum Sudan grass. These are 2 good varieties. Extra Grays, Evergreen Seed used to sell Extra Grays. That's still available, and it's sold by a company. It's produced by a company called Coffee Forage Seeds down in Texas, where they grow all these summer annual seeds. That's been a pretty good sorghum Sudan grass variety over the years. So there's 3 good varieties that you might consider putting into a forage program. All right, I want to change gears a little bit and talk about some work we're doing with silvopastures at the research station. And this has been pretty exciting. You know, last week I went to southeastern Virginia to visit a farm with the extension agent that's thinking about putting a silvopasture in. And just in that neighborhood, there's 2 or 3 farms that have incorporated silvopastures into their grazing systems. I think they've got some real benefits in terms of providing shade for livestock during the summer months, if they're done right. So what is a silvopasture? And it may be easier to answer the question what it isn't. It isn't new. It's been around a long time. May have gone by different names. It's not just turning cattle into woodlots. And we have a lot of that. I call it woodland grazing. But that's not really a silvopasture. Silvopasture is something that's managed. It's not a single tree in a pasture. Everybody in here knows what happens to a single tree in a pasture. They congregate underneath it. It's a nutrient magnet. They go there, they lay, they ruminate, they get up, they urinate and defecate there, and over time they concentrate nutrients there and kill the grass underneath that tree. What silvopasture is, it's a sustainable practice. It integrates trees, grass, and livestock into a managed system. And the key word there is managed system. So we're just not turning these calves in there or cows in there and leaving them. We're managing that grazing. We're using rotational stocking underneath those trees. And that's important. One of the neat things about silvopasture is it has two economic time frames. So the short-term time frame would be returns you get from livestock grazing underneath that. The long-term time frame would be harvest timber that would become marketable at some point in time. Probably the biggest single advantage I see for cattle producers in Virginia in terms of silvopasture is shade during the summer months. You know, you can say cattle don't need shade, but man, when I look at cattle in the summertime, they got a black coat on and it's 95 degrees out and the relative humidity is 90%. They're suffering. And getting some shade, getting them in some shade is really a good thing during the summer months. And I think that's going to be a big positive of silvopasture. And you don't want to plant your whole farm to silvopasture, but having some paddocks that you could go to in the summertime for shade is going to be pretty important. So we got this grant from the NRCS. It was a Conservation Innovation Grant. And at the research station, we're demonstrating the establishment and management of silvopastures. And if you haven't been down to look at it, I've kind of been featuring on field days and tours so that you can watch it as we kind of go along. And I'll show you where we're at now with the project. But what's kind of neat is that we put together a team of different people. And there's all kinds of people from different agencies there, from the Department of Forestry, from NRCS, from another university, and they're all there to plan this project out and give us their input. And what I found out is there's a lot of unanswered questions about how to actually establish silvopastures in this part of the country. So our objectives were to demonstrate the establishment of pine and hardwood silvopastures, to demonstrate the use of managed grazing in silvopastures, which I think is going to be really, really important for the success, and to provide educational opportunities for all those interested in silvopastures. Look, if you're driving through Blackstone and you'd like to take a look, stop by and see me. I'd be happy to take you out and show them to you any day. So we've got this is our map of our silvopasture area. There's 40 acres in this demonstration area. And what's kind of neat about this demonstration area, it's on the research station of Blackstone. Half of it is in hardwoods over here, and half of it is in pine plantation. So we were able to kind of just break this in half and demonstrate both hardwood and silvopasture and pine silvopastures. Now, you know, this has to have a little bit of a research along with the demonstration. So what we did was we laid out a sampling grid here. We've got 96 permanent sampling points established in here. That's about one sampling point per 0.42 acres. And before we cut the first tree in this silvopasture, this is an established stand of pine and hardwoods, we went through and did an intensive sampling to try to get a snapshot of this ecosystem before we did anything. And then we're going to come back after we get silvopasture established and take another snapshot at these same points. And I'll show you some of the data that we've collected to see how this ecosystem changes as we move from a forest into a silvopasture. So we looked at a whole bunch of different things, insects and macroinvertebrates like earthworms and centipedes, and we looked at all the traditional soil chemistry, and we looked at soil microbes, looked at bacteria and fungi and bacterial to fungal fungi ratio, and soil carbon and nitrogen and leaf litter on the ground, and tree community and soil nematodes. And we actually dug soil pits and went through the soil looking for different invertebrates in the soil. So just a whole bunch of things. We tried to get a good overview of that whole ecosystem before we did that. And it was a lot of work that first summer. And this is Paul Sempner. Many of you that grew tobacco know Paul's our entomologist. He's retired now, but he's also a great insect ecologist. I mean, probably better insect ecologist than a tobacco entomologist. And he came back and is helping us with this project. And Paul just loves insects, and we were happy to have his help. We're sampling daytime flying insects, and he sampled nighttime flying insects with light traps and ground-dwelling insects. We actually took samples of the leaf litter, and actually, it's tedious, but we went through each sample and picked out the insects that we found in that leaf litter layer in there. And then we actually went in and dug soil pits, one cubic foot of soil, which doesn't sound like a lot, but it's quite a bit when you have to go through it. Actually took that soil and went through it and picked out the earthworms and the insects and anything that was a macroinvertebrate in that soil. And we've got all this documented, and I'll show you some initial results. The other thing that we did was we did a very detailed survey of the timber stand before we started. We had some forestry students come down from Virginia Tech, and they did a cruise of that, a very intensive cruise of those two stands. And they came up with a whole bunch. I'm not showing you all this, but the kind of species that were present, the kind of understory, epicormic branching on the trees, how much timber was there, the basal area per acre, and so forth. And the predominant tree species were oak species in our hardwood area at about 120 square feet per acre basal area. That's if you cut that tree off, that's a stump area. And then loblolly pine had quite a bit more, 382 square feet per acre of basal area. And we did soil chemistry, and as you would expect in a timber stand, our pH was low, about 4.9, so that's going to need about 3 to 4 tons of lime to bring it up to a level that's going to support grass growth. Very, very important. If you're going from a timber stand into silvopasture, you're going to have to adjust soil fertility. Phosphorus was very low, as you would expect, 2 parts per million. That's in the very low category, and we're going to have to add a significant amount of phosphorus to these systems. Potash was in that low plus medium range for soil test range, and then we kept nitrogen and carbon estimates there too for the soil. Total microbial biomass was estimated at about 28,000 pounds per acre. That's incredible when you think about it. That's things like soil bacteria, soil fungi, protozoa, and other microorganisms in the soil was there in large quantities in this forest soil. And they tend to be dominated by a fungal species in a forest area. And that'll probably change to a bacterial species dominating that system as we move into pastures. But we don't know that for sure, so that's one thing that we're going to track is that bacterial to fungal ratio. 15 different species of nematodes were found. Nematodes are microscopic worms that live in the soil. Some are bad, like in tobacco and soybeans, and some are good. Our most abundant insects were camel crickets, pill bugs, harvestmen, millipedes, and field crickets. So after we got all this done, we came in and we worked with a professional forester. We told him how much area, basal area, we wanted to leave per acre. We wanted to take it down to 50%. So in our pine stands, we left around, we started with 390, so we left about 180 square feet per acre, and about half of what was in the hardwood stand also. So he came in and he marked trees with blue paint. He did an amazing job. We came very, very close to our target of 50%. I don't know how he did it, but he did. And then we worked with the forester who came in, and they weren't overexcited about having a bunch of blue trees in there that they had to leave, but they did a good job of not damaging the trees that were remaining. And we watched them pretty close to make sure there were no trees with blue paint going out. So, and they did a good job. And that's kind of what the stand looks like now. And what we're doing this winter is that we're amending the soil, and we're getting ready to plant the grass this spring.
What's the tree age?
You know, I haven't counted the rings. I would estimate they're probably somewhere between 25 and 35 years old. The hardwoods are older. The pines were replanted somewhere along the line in there before I came. So one of the issues that you're going to have with the silvopastures is stumps after you're done. Even if they cut it low, everything kind of sinks down a little bit after you harvest the forest, and you got these stumps there, and that's really hard on equipment. So what we did was we came in with a mulcher, and you guys, I think Scott actually had a mulching demonstration several years ago at one of the field days. And they came in and they mulched all the stumps down to the soil surface or just a little bit below it. Is there any questions on the silvopasture? All right.
What's the effect going to be with, like trees want that pH of 4.8 to 5, and your pasture, you want that pH of 6. How's that going to affect trees? Like after the first harvest, are you going to be able to, say, plant loblollies, replant those loblollies back?
You know, I don't know the answer to that. I'm not a forester, but from what little I've read is that the loblolly is it doesn't have to have acid soils. So if it's a little bit higher, I think it'll still be productive in there. One of the changes will be the phosphorus availability in that soil. As we lower that pH, that phosphorus becomes much less available in that soil, if you've ever seen those graphs with nutrient availability. And it gets bound up. So increasing the pH and increasing the fertility may actually enhance tree growth over the long term. But that's one of the things that we'll be tracking is how these trees change as we implement the silvopasture practice. All right. The last thing I want to talk about tonight is just a little bit on tall fescue. Tall fescue is what we've got to work with, and we're doing a pretty interesting project right now that's looking at animals that have some resistance to tall fescue, toxicosis. And I'm going to tell you right up front is that this is not a silver bullet by any means. But there are animals within herds of cattle that you can tell just do better on tall fescue than others. And this is a picture. You can kind of see this calf here. This was from a group of calves that we had this summer. This one here, all the way in late summer, we spent the whole summer on novel endophyte tall fescue, and it just did not slick up. It had that shaggy hair coat all the way in late summer. And you see calves like that, and this came off of hot toxic endophyte infected tall fescue. And then there's a calf right beside it you can't see well in this picture that just slicked up nice. So there's something going on genetically in these animals. And there was some work done at Tennessee several years ago. It was just published last year that showed 2 genes that they've identified so far that are related to increased tolerance to tall fescue toxicosis. DRD2 and then one other gene. And so for this project that we're working on, we went through and we sampled the brood cows at our Shenandoah Valley Research Station. We sampled 161 cows. 30 of them had AA gene configuration, which was resistant to tall fescue toxicosis. 80 had AG, which was partial resistance. And then 51 had no resistance, which was pretty interesting. But it got even more interesting when we sampled their calves, and these are the calves that we had at the research station this summer. If you remember, 14 of those cows had this resistance, 30, and then only 14 of the calves had it, which was pretty interesting. 14 of the calves out of that 160-some calves had resistance to tall fescue toxicosis. And you're probably saying, well, what's going on here? Well, at our Shenandoah Valley AREC, we have a good artificial insemination program. We use good bulls that are selected by someone at Virginia Tech, I guess. And sometimes we select bulls that may have been raised outside of the fescue belt, maybe from Colorado or Nebraska or somewhere, and have never seen tall fescue before. Those bulls have good traits. There's no question about that they're bringing to these calves. But but resistance to tall fescue toxicosis may not be one of those traits. And what was really interesting when we went through and looked at who sired these calves, these 14 calves, it was cleanup bull. And you know where that cleanup bull was from? South side. So something's going on genetically with these animals. And so what we did for the study that we're working on, this is kind of a unique study, is that we kept back 12 of these 14 calves. And this winter, they're getting a cannula in the side, which is a big rubber, a hole in the side of the calf with a big rubber stopper in it about 7 inches in diameter. What we're going to do next year is we're going to take these calves. We've kept 6 that had resistance to tall fescue toxicosis and 6 that did not. And I'll tell you this, when I selected those calves, we did it on a genetic test that was done in Tennessee, and I did it on paper in my office on the computer, and I said, we're keeping these 6 and these 6. We went out to the field, we sorted them out of 75 calves, and you could look at those calves and you could pick the ones that had resistance out just by looking at them. And an agronomist can do that. Any of you in this room could do that, because the ones that did not have the resistance were shaggy. I mean, they just looked rough. And the ones that had the resistant were nice and slicked off. It's pretty amazing. So we've got these calves. We're going to put a cannula in them, and then next spring we're going to graze toxic tall fescue, and we're going to take samples of the rumen fluid, and we're going to look at what happens to the microbial population within the rumen as impacted by novel endophyte or toxic fescue, and then we're going to look at the genotype of the calves. Do the genotype that's resistant have a different set of microbes in the rumen versus the ones that have the resistance? So we don't know. Nobody has ever done this before, so we're pretty excited about this study. And I can't do that. I'm working with a biochemist at Virginia Tech. His name's Bisharu. I can't even pronounce his last name. But he's the kind of guy that you stand beside and you just kind of feel dumb. You know, he's just so bright. And he's going to do the metagenomics on the DNA of the microbes in the rumen to compare the 2 different populations. All right, and that kind of brings me to the last thing I wanted to mention to you all tonight, and that's this winter's Forage and Grassland Council conferences. These are going to focus on tall fescue in the 21st century, understanding and managing tall fescue and grazing systems. This is going to be the best tall fescue conference in the country this year. I guarantee it. And it's going to be right here in Virginia. If you've got cows and you've got tall fescue, you need to come to this meeting. There should be a handout at your place. This is going to be a great meeting. I mean, we've got world-renowned speakers on tall fescue coming to Virginia to spend the week with us. So please come to this meeting. It's going to be in Blackstone, probably be your closest location, and spend the day with us. I know you're not going to be disappointed. And one of the speakers is coming from the University of Missouri, who's commercializing a genetic test for animals for tall fescue, resistance to tall fescue. Any general questions?
I want to know if it's going to snow that day.
Carolyn, I hope not. I know she's been trapped in Blackstone with snow.
I'm telling you, it's miserable.
Yeah. We actually buy snow insurance for those meetings, believe it or not. Those meetings cost a tremendous amount of money to put on, and if one ever got canceled, we'd be in big problem. If you look on the inside here, it says it's got the program. Registration's at 8:30, and it ends at 3:30, and lunch is provided. All right. Thank you all for your attention. I really appreciate it. Thanks for having me back again.